Okay, we're going to do a simple demonstration on DNA replication, or how DNA makes a copy of itself. We're going to just use some pipe cleaner and beads, and it's important for you to remember the base pairing rules for DNA. So the key here is that A always pairs with T and C always pairs with G. A is represented with a red bead, T is a green bead, C is a yellow bead, and G is a blue bead. And as you can see, I have them paired together down here. Okay, so the first thing that needs to take place in order for DNA to make a copy of itself is it has to get untwisted or unwound, commonly known as unzipping DNA. It's got to have some help to do this. It uses an enzyme known as helicase to unzip itself. Okay, the DNA is now unzipped with the help of the enzyme helicase, and it has created what's known as a leading strand and a lagging strand and a replication fork. So the leading strand is going to be this top strand here. This is the lagging strand at the bottom, and then this area right here is called the replication fork, and that's as far as helicase has unzipped the DNA. On the other side of this, the DNA is still zipped up. So the leading strand is going to be replicated in a 5' to 3' direction, and the lagging strand is going to be replicated in a 5' to 3' direction. You can see they're opposites of each other. You'll see that we will replicate the leading strand in one continuous replication. Now the lagging strand is going to have little segments replicated at a time, and those are called Okazaki fragments. Okay, so we're just going to concentrate on that top leading strand right now. And again, remember, it's going to add complements in the 5' to 3' direction. Now, it needs help to add those complements, and that's done by what is known as DNA polymerase. So DNA polymerase is going to come along and read these bases so that it can figure out which complement bases it needs to get to replace it. So as it reads, it fills in, and then it creates your complements. And so here you have a complementary strand of DNA. You can see our original strand was T, which would be a complement would be A, and then we had a G, the complement would be C, and so on as we move down the strand. Now we'll move on to the lagging strand. Okay, now focusing on the lagging strand, or the bottom strand of the original DNA. This one's done a little bit differently. First of all, we use a RNA primase that's going to begin reading the strand and making an RNA primer so that DNA can then be laid down. Then polymerase is going to come in and read the strand just as it did with the leading strand. And it's going to place the complements, but this is going to be done in fragments. Again, those fragments are known as Okazaki fragments. Now once it has the fragments in place, it's going to use another enzyme to help put those together and fill in those little fragmented sections, and that is known as ligase. So we have three important enzymes in DNA replication. First one is helicase. It unzips and unwinds the DNA. Second one is DNA polymerase. This is what's going to read the leading strand and the lagging strands in order to put those complement bases down. And then finally we're going to have ligase, and it's what is responsible for filling in the gaps in those Okazaki fragments. Now once we have the leading strand and the lagging strand complemented, we then need to again wind or twist and zip up those DNAs, and that's going to be done by helicase. Okay, before we use helicase to zip these strands back up, I want to show you that if you look, this was our top strand of our original DNA. And if you look, you'll see that that top strand of the original DNA is the exact same thing as our complement strand that we made for our lagging or bottom strand. And our complement strand we made for the leading strand is exactly the same as what our lagging strand was. So there's one piece of original DNA in each new DNA copy, and one new piece in each new DNA copy. Okay, so let's use helicase to twist these back up. Okay, we're all done. We've used helicase to zip it back up, and as you can tell, we have 2 exact copies of DNA. All your cells have these exact copies of DNA that are made through the process known as DNA replication. Our next section will be protein synthesis.